Today I would like to introduce a therapeutic concept that came from looking at inflammation from a slightly different angle. In many hard-to-treat diseases, the problem is not only that something harmful enters the tissue. The problem is also that damaged material cannot leave the tissue efficiently. In other words, pathology is often driven not only by what enters the tissue, but by what cannot be cleared from it. The innate immune system is designed to protect us immediately, but under certain conditions, this protective response becomes persistent, self-sustaining, and damaging. I refer to this process as innate protection-related collateral damage, or IPRCD. The therapeutic idea I will present today is not simply to suppress inflammation. It is to restore the tissue's ability to process, neutralize, and clear pathological extracellular material. Many different insults can initiate local tissue damage. It can be infection. It can be ischemia-reperfusion injury. It can be toxic exposure, trauma, or sterile tissue injury. At the beginning, these conditions may look very different, but biologically, they often converge on a similar innate immune response. The tissue recognizes danger. Neutrophils and other innate immune cells are recruited. A local containment program is activated. This convergence is very important. It means that even though the triggers are different, the downstream failure mode may be shared. The key bottleneck is not always the original trigger. Very often, the bottleneck is failed resolution, the inability to remove damaged extracellular material without generating more damage. This creates an opportunity for a common therapeutic strategy across multiple diseases. The innate immune system does not begin by asking how to fully repair the tissue. It begins by asking how to contain the threat. This is why many early protective responses are structural. One of the clearest examples is the neutrophil extracellular trap, or NET. NETs are DNA-based extracellular structures decorated with histones and granular proteins. They are highly effective at trapping pathogens and localizing danger. At the initial stage, this is productive. The system limits spread and buys time. But the same structure that protects can become harmful when it persists. NETs are not inert. Their components, especially extracellular histones and DNA protein complexes, can be cytotoxic and pro-inflammatory. So the biological problem is not containment itself. The problem is containment that persists without adequate clearance. The transition from protection to pathology begins when the containment matrix is no longer cleared efficiently. NETs and similar extracellular matrices are gradually degraded by endogenous nucleases and proteases. But degradation alone does not guarantee resolution. Instead, degradation can generate toxic fragments. These fragments injure surrounding cells. Injured cells release new danger signals. These signals recruit more neutrophils. More neutrophils generate more NETs. At this point, the system enters a self-amplifying loop. The original trigger may already be reduced or even gone, but the tissue remains locked in a pathological cycle driven by its own protective machinery. This is the key definition of IPRCD. The disease is no longer driven by only the initiating insult. It is now driven by collateral damage from a persistent innate protection program. Once this pathological state is established, it is maintained by three coupled drivers. The first driver is diffusion block. Persistent extracellular matrices create mechanical and physiochemical barriers. They impair diffusion, convective flow, drainage, and access for therapeutic molecules. The second driver is cytotoxic fragments. As the matrix is degraded, it generates fragments that can directly damage cells and amplify inflammation. The third driver is inflammogenic clearance. If toxic debris is handled through complement Fc receptors or other inflammatory pathways, clearance itself becomes a source of amplification. These three drivers reinforce each other. That is why single-target therapy is often incomplete. If we only degrade the matrix, we may improve diffusion but increase toxic fragments. If we only neutralize toxicity, the matrix and clearance problem may remain. If we only suppress inflammation, debris may persist. A coupled pathology requires coupled resolution. This leads to the central therapeutic design principle. If pathology is maintained by a coupled system, then effective therapy must also be coordinated. Resolution requires at least three functions. First, transport has to be restored. The pathological matrix has to be opened or gradually disassembled so that trapped material can move. Second, toxic fragments have to be neutralized. Otherwise, matrix degradation may simply convert one problem into another. Third, the resulting material has to be routed into non-inflammatory clearance pathways. This is the critical point. The goal is not just to break down extracellular pathology. The goal is to control what happens after the breakdown. In biology, degradation is not the end point. Clearance is the end point, and the quality of clearance determines whether the tissue moves toward resolution or toward further amplification. The therapeutic strategy is to convert a pathological matrix into a neutralized, clearable cargo. This can be understood as a sequence. Matrix becomes fragments. Fragments become cargo. Cargo is routed toward physiological clearance. Each step can be therapeutically controlled. DNase can be used to disassemble DNA-rich matrices such as nets and help restore transport. Clusterin can bind toxic net fragments, including histone-containing material, and reduce their cytotoxicity. But clusterin's natural clearance pathway appears to be capacity limited. Therefore, the next step is to increase clearance potency by redirecting clusterin-bound cargo toward a more efficient silent clearance route. One attractive option is TAM receptor-mediated clearance, using routing domains such as PROS1 and GAS6. So the therapeutic objective is not degradation alone. The objective is controlled cargo handling. Disassemble when appropriate, neutralize what is released, and route it towards non-inflammatory clearance. This brings us to the platform opportunity. Many current anti-inflammatory approaches focus on suppressing signals. That can be useful, but it does not necessarily remove the pathological material that keeps generating those signals. Our approach is different. We are not primarily trying to suppress inflammation. We are trying to restore clearance capacity. The broader therapeutic category is control of extracellular clearance. The platform may be relevant to net-driven inflammation, sterile injury, thrombotic and fibrotic pathology, and potentially amyloid-related diseases where extracellular persistence and failed clearance are central features. The conceptual shift is simple. From persistence to resolution, from amplification to control, from pathology to homeostasis. The platform is early and patent pending, but the leverage is high because it targets a shared failure mode across multiple diseases. Thank you.